gives me a great pleasure and a real privilege to introduce Dr. Tom Insel to really help summarize the day and the afternoon for us in a capstone speech here at the DECODE Summit. Tom Insel is the Director of Clinical Neuroscience at Verily, the life sciences company that's part of Alphabet, formerly Google Life Sciences, where he's working on developing the group's medical technology efforts, particularly within the area of mental health. Dr. Insel, as most of you know, served as the director of the National Institute of Mental Health, NIMH, for a long period from 2002 to 2015, overseeing advances in mental health, neuroscience, diagnostics, and therapeutics. Prior to being at the NIMH, Dr. Insel was a professor in the Department of Psychiatry and director of the Center for Behavioral Neuroscience at Emory University School of Medicine. He also serves the director of the Yerkes Regional Primate Center. Tom is best known for research during those years on oxytocin and vasopressin, two peptide hormones implicated in complex social behaviors such as parental care and attachment. Dr. Insel is also a member of the National Academy of Medicine, National Academy of Sciences. He's a recipient of numerous awards and accolades. I won't go through the list. Most notably also as a BBRF Productive Lives Award recipient in 2014, and the Autism Science Foundation Distinguished Scientist Award in 2015 in the more recent years. Tom, it gives me great privilege to introduce you and to have you here on stage.
I wasn't even sure what a capstone speech was, but I did Google capstone, and that's what I came up with. So a capstone is what goes into the arch before it's finished. Unfortunately, it's supposed to support everything, which I'm not prepared to do, but it will maybe be useful to tell you what led to the arch. A lot of the ways that I think about where we are and where we have to go were presaged by a mentor of mine, Sidney Brenner, who said this many, many years ago, that progress depends on new techniques, new discoveries, and new ideas, probably in that order. And that's actually a lot of where we've been in the course of the day. In fact, if you think about it in a way, Mark started us off with this comment at the very beginning, and he actually said three things. It was quite interesting. He said, look, we're going to need some new approaches, right? So we've been very focused on the technology that we've got here, but we've got to change the questions that we're asking. And then he said, so this is like on Sidney's second comment, in terms of discoveries, it's going to be really critical that we start to think about the analytics side of this. And actually, I was surprised we didn't spend more time talking about this today. But creating the models and creating, as he said, sort of unsupervised approaches to the raw data that are coming out from optical imaging, a topic which I think may deserve now its own meeting because it's a really big and important and I think urgent topic. And then he also said, as we've been talking about more recently, the need for us to say something about how this ecosystem can change and how we support that. So let me review with you quickly what actually happened after Mark's comments. We had the panel on circuits, which I thought was great, and we heard from lots of people about where the challenges are. Anirvan reminded us that there's like no real link between the genetics and the circuits yet. And Corey pointed out about how circuits are really whole body things, but we can learn a lot from comparative studies. Guoping, as expected, said we really ought to go back and thinking about how these things are built in development, and that will really be the key. And Tim Dennison reminded us that, as Mark had said at the beginning, we're probably going to need even better devices if we want to do this right. We're going to need better technology. Mark Baer was the sort of leadoff hitter for the keynote talks, and Mark, I think, surprised all of us. We expected him to talk about Fragile X. Instead, he kind of threw us a curveball and said, well, you know, Fragile X doesn't actually work all that well in going from genetics to circuits. In fact, the targets are going to probably, the therapeutic targets are probably not going to come from the genetics, but from the physiology. And then he shifted to explaining how schizophrenia might be a really interesting new model looking at this failure of habituation. And I thought provided us a really interesting approach around NMDA receptors. Garrett picked up on a piece of that around this PNOC, which I actually didn't know much about, but I think it's a really cool story to suggest that the power of being able to get to cell-specific intervention, cell-specific understanding of not maybe an illness, but of a behavior. And in this case, picking up on one of David Anderson's comments, really going after the behavior of anxiety. And I think he gave us a really great picture of how one would do that using Inscopus tools, using optogenetics. And then what I thought was maybe one of the most exciting things of the day, sort of saying, all right, that gives you some idea of what cells might be important, but if we really need to, what we really need to do is to get to the single cell level. And by bringing in DROP-seq gave us a great opportunity, I think. If we do this next year, you've got to come back and tell us what you found, because I think that's really the level that we ultimately want to get to, where we're being able to identify what's different about the 10% of cells that really seem to be critical for the plus maze behavior. Jones took us, I think, a long way into the same kind of an approach for understanding Parkinson's. And I think if, Inscopus is looking for a kind of a case study of where the technology is really helpful, this is a beautiful one, and maybe one that the public needs to see more of. Because, yes, we had a model of how Parkinson's works, we thought, but I think actually it's now really compelling. I think that what Jones did with the D1, D2 story is for the first time showing that this isn't just a model. This is actually the way it works. And we can recapitulate this with great precision. We can treat it and look at how interventions make a difference. This is a very powerful example of where the technology gives you the discoveries and now can help us to think about new concepts. Dr. Okano sort of blew my mind on in a couple of ways. One was how rapidly the marmoset work is going forward, and and it's kind of astounding that you within a couple of weeks you could deploy the Invista technology and already have fresh data, which is kind of wonderful. What I was really struck by in that presentation was the ability to, for the first time, to go after a problem that has really stymied us. So in Parkinson's disease, all of our work has been around the late stages of when you've already lost all of your dopamine cells. We now know how to identify the prodrome from the sleep disturbance, from constipation, from the anosmia. We've got now a very well-defined set of symptoms 10 years before you begin to develop the motor symptoms. We have had no way of studying that and beginning to do clinical trials in that arena. It's very, very hard to find it and to validate it. And it's amazing if indeed a marmoset will show you the changes in sleep, in REM sleep, before the motor changes. That is just an extraordinary opportunity for the first time to do the most important interventions for Parkinson's, which would be, of course, to prevent the loss of nigral cells rather than to just try to replace dopamine once they're gone. So that's an incredibly exciting advance and something that I think will be fun to follow. We broke for lunch, came back, and heard an extraordinary story about how the visual system, how the retina works, and a story around starbust cells that from Botan Roska, which is, again, just elegant work of using retinoids and even now combining retinoids and organoids to begin to actually look at the whole story of what ganglion cell axons are doing and how one particular, it's an amazing example of a single gene in a single cell having a very discrete effect, in this case, on direction selectivity. So just elegant example of what one can begin to do with cell-specific interventions. Finally, Helen took us through sort of the other side of the coin, which is like one of the most complicated and certainly both in terms of genomics and in terms of the circuitry, complicated disorders around depression, and helped us to realize that even here, if we think about it in the right way, we can make real progress clinically. And she kind of teed this up at the beginning of the day with a question for that first panel about, well, do we really need to understand everything to be able to intervene? And can we sometimes do this in the way we don't actually understand everything about atrial fibrillation, but we are able to put in a pacemaker and be able to ensure that we can normalize cardiac rhythm. And is that really the way we should be thinking about these disorders as arrhythmias that give us an opportunity for interventions, even when we don't fully understand what's driving the arrhythmia? So we had a second panel, which then got into the ecosystem issues. Make sure I didn't, and there, I think we don't have to think back too far to remember what you heard there. Most of this, of course, was talking about what we then later went into with the different tables, kind of wrestling with what we can do in terms of funding, in terms of partnerships, where the opportunities are for progress by just changing how we do the science, not only what the science is that we do. I'm not going to go through this list. You can see it. But I do think that what we can take from that was a sense that I think from almost every stakeholder in the room, we can do better than what we're doing now. And we shouldn't assume that what we do now is acceptable. So we really do need to think about how to change the culture of what we do to be able to get more progress. So I'll just summarize to say that if we go back to Mark's original charge to us at the beginning of the day, where he said we need new approaches, we need analytics, we need to look at the ecosystem, what I heard was that on the new approaches, we could do a lot better in integrating across levels. And again, you just heard this from David and can't remember, maybe it was Guoping or no, it was Hong Kui at this table who said, we can put all this together in a way that's probably much more effective. We didn't talk much about it. My own bias here is that one of the places we could do much better is simply by having much better descriptions and much more objective and quantitative descriptions of behavior. When I was at NIMH and really pushing the study of genomics and neuroimaging, David Anderson used to write me these nasty notes about how we need to do a better job of just studying behavior and cognition. And I would, because I was then director, I would be very polite and say, thank you for that suggestion. David's left, so I guess I don't have to, oh, shit. I now actually have begun to listen to him, and I actually think we have the tools to do this really well in a way that maybe we didn't have the tools so much 10 years ago. I think we have the tools in mice for sure, but we have the tools in humans. And so I would encourage that one of the things that we should think about going forward is not just biomarkers, but what we now call digital phenotyping, or finding ways to do far more continuous and precise measurements of human behavior, everything from motor behavior to sleep behavior to mood and activity, and that we are able to do because we have technologies that help us do it. We also heard about the need for some very large studies to track people over time, and I think that's certainly something that's paid off well for ADNI, in the Alzheimer's field, and we could probably redo that, but maybe with some lessons learned from ADNI in many other areas. I would suggest that if we're going to do it, maybe we don't do it disease by disease, but we think about a broad population approach, since people don't really, at least in the psychiatric space, almost ever have a single disease, and when they have a single disease, it almost never matches perfectly with the diagnostic criteria anyway, and we don't really know what the boundaries are for any of these diagnostic groups. So to think much more broadly about psychopathology and to think about functional outcomes and not just symptoms, which is another point that has come up a couple of times during the day. On the analytics, I'm going to harken back to Mark's original point. I don't think we've talked enough about this, but I do think as we go forward, there's an opportunity to take what's happening in machine learning, and I'm in a company like mine, This is what we live and breathe every day, and it's changed dramatically in the last 6 months. We are not where we were in December or January. This is a completely new, very fast-moving, very powerful field. It will be changing profoundly every area of science that has to do with pattern recognition. And when I see those optical imaging videos, I just find myself thinking, wow, what a fantastic opportunity if you want to try to decode brain signals. Why aren't we bringing the very best machine learning to those kinds of data sets? And could we get enough of them together to begin to really rock and roll on that? I think that would be a project that would get a lot of engineers very excited. We talk a lot about neural nets as a kind of model for machine learning. That's sort of nonsense. What we really need to understand is how the brain computes, and then to begin to have brain-inspired computing. And I think we could get a lot of people excited from the tech industry in working with neuroscientists who have that kind of data, who have the data of individual cells dynamically over time in a behaving animal, where you can begin to say, all right, how is the information encoded? How is it being processed? How is it being stored? Those are all the problems we think about in building the next generation of computational machines. Why don't we try to do this knowing a lot more about what the brain does? And to understand what the brain does, we're going to need to have the kind of machine learning that people are doing elsewhere, but not yet, I think, in this area. And then finally, nothing really that you haven't heard already on the ecosystem, except that, goodness, let's try to fix this. This is, somebody once said to me at NIMH, we have everybody in the room we need to make this happen. We don't really need to ask anybody else to do this for us. And I think that may be the case here. This is a fantastic group that Inscopix has brought together, and there really is an opportunity with the people in the room to try to change the world for this particular set of problems. Go back, actually, it was so interesting to hear the reports out in this last session, and it was the trainees who had sort of the most ambitious vision of what might actually be built here, and how wonderful to maybe just turn it over to them and let them tell us how we should do this. Which brings me to my last slide, which is that Bill Gates once said that we always overestimate the change that will occur in the next 2 years and underestimate the change that will occur in the next 10. I know the 10 years is like 2 R01s, I guess, in NIH time, which may sound like forever. But the reality is we probably do need the trainees who have a 10-year horizon to take all this on and really help us all think about how best to change both what we do, which is the science, and how we do it, which is the culture. So with that, I want to thank Inscopix for bringing us all together. Kunal, Pushkar, you guys have done a spectacular job, and all the team that has worked around you. It's been a wonderful day, lots of great ideas, great interactions, and I think we should give all of you a round of applause. Thank you.